Hi, I am Mrs. Sloan, and this is a video intended for my honors biology students. And this is all about control of gene expression. So this is video one. And let me make myself a little small. Here we go. This is video one. So we're going to talk about prokaryotes and eukaryotes and how their genes are controlled. And then part two, the next video will all be about the failure of control, and that is cancer. So if you're new to my videos, down in the descriptor of the video are the notes that my students use, which you are welcome to use. They are two columns. Column one, I'll help fill in the scaffolding of the notes, and column two, you should put in pictures that might be helpful for you. All right, so we are on chapter 25. I want to just do a quick review of a couple of things, right? You know this. So DNA to DNA would be replication, right? DNA to RNA is called transcription, and then using that RNA to build a protein is translation. And so we're going to talk about levels of control all along there. So all of our cells in our bodies have the exact same DNA in them, right? Every single cell has the exact same DNA, yet our cells are so specialized. So how does that happen? Well, the key is differential gene expression. So you have certain genes that are turned on and certain genes that are ignored. And this happens at different times in our life and for different lengths of time in our lives. It can vary throughout the day. It can vary throughout the month. So in general, the first thing you need to put in your notes, in general, number one, all cells have the same DNA. How do cells become specialized? Differences in gene expression, and genes are turned on and off at different times and in different cells. So regulation requires multiple steps, and it can happen at any one of these steps. It can start with the DNA. It can start with the process of transcription, how it's the process of transcription making RNA, how that RNA is processed, whether or not it can get out the cell, how long that mRNA lasts, and how long it is able to be translated, and even the folding of the proteins. Now, this assumes that you are a eukaryotic cell. And for our first discussion of control of gene expression, we're going to talk about prokaryotes. So how are they different? They don't have a nucleus, right? They have a single circular chromosome. So we're going to look at some examples for that. So on your notes, the introduction, okay, is all about prokaryotic regulation. And you need to know that prokaryotes, let's say they do live in our gut. And so they can adjust what proteins they need, what enzymes they need based on what we're putting through our intestinal tract. And so they'll synthesize what they need when they need it, and they don't want to make it unless they need it, right? It's kind of like if you think about if you go in a room, you turn the light on in that room because you need it, because you're in the room, but when you leave the room, you need to turn the light off, otherwise it's wasteful, right? And so bacteria's environments are always changing, so then are their necessary proteins. That's number one underneath it. Okay, so now let's start talking about how bacterial cell gene expression is controlled. So they have what's called operons. So take a look. First of all, let's get our bearings here. Can you see the double helix of the DNA? Okay, and you see where these slash marks are. I need a pointer. Here we go. You can see where the slash is right here. That means it's farther upstream. It's the regulator gene is not right next to the promoter. Okay. Now, if you remember when we talked about transcription, we said RNA polymerase binds to the promoter. So that's where it docks on, and then it would transcribe these structural genes. Well, here you can see you have an active repressor, and that active repressor is bound to something that neighbors the promoter, and that's called the operator. And do you see this particular repressor, how it kind of extends out over into the promoter? So this inhibits RNA polymerase from binding and also from moving down this gene sequence so it can transcribe it. Now, where did this repressor come from? Well, farther upstream in the DNA, you have a regulator gene. When that regulator gene is transcribed with RNA polymerase, right? So it would have its own promoter, right? When it is transcribed and translated, the protein that you build is this active repressor. Now, I'm going to be talking to you here in Honors Bio about a repressor, like it's good to go boom. As soon as it's built, it starts inhibiting this reaction. But there are other repressors that are built and they are inactive and they have to actually be turned on. All right. So this is not the only way that it is done. So on your notes, you have an operon as in bacteria. Genes are often clustered into units. And then this is the kicker right here, which allow for the expression of several related genes to be controlled as a unit. Okay, to be controlled as a unit. Now, you don't call an operator now, right, on your phone. And most of you have, cell phones. You may not even have a landline anymore. But I need to tell you where this comes from. Okay, in the 1950s, Jacob and Nod, when they discovered this, an operator played a different role. You literally would call a town and an operator would pick it up and you would say, I'd like to talk to the Jones, please. And then she would connect you to the Jones. And if you didn't have that operator there, if she was somehow repressed, then you couldn't talk to those people. So this is showing you that they use this as a model, using the term operator to explain this critical role. Because if this operator is repressed, then there's no way these genes can be turned on. And in this state, this operon is off. All right. So now let's take a look again at another drawing, but see if we can apply what we learned. Here's that regulatory gene farther upstream. If it is transcribed here into this mRNA and translated, you have this active protein that binds to the operator. And because of that, RNA polymerase cannot get on the promoter and you cannot transcribe those genes. Now, that's not a bad thing. It's kind of like you don't turn on the lights in a room if you're not there. If you don't need these genes transcribed, don't waste your time and energy building the proteins that they code for. But if you do need them, you want to be able to turn those genes on. How might that happen? Well, this repressor will remain here unless, and get your bearings here, here's your regulator gene, active repressor, RNA polymerase is not on the promoter. This operon is off. And the reason is these genes, if you transcribed them and translated them, they would build a protein that digests lactose. If you don't have any lactose present, you don't need those genes turned on. But if lactose is around, here's the cool thing. The lactose itself, which is a disaccharide, that's why it's represented as two spheres here, binds to the repressor. And when it binds to that repressor, it removes it, right? Because the repressor can only bond either to the operator or to the lactose. So if it binds to the lactose, it changes its shape so it can no longer bind to the operator. That's good because we need RNA polymerase to bind to the promoter and transcribe these genes. And when it does, it builds the enzymes that will digest this lactose. Well, that's perfect, right? Because once all the lactose has been digested, you don't need these anymore. And so once the lactose has been digested, it's no longer able, it's not around to bind to the repressor. And so the repressor becomes active again and shuts it off. Okay. It makes sure you only transcribe those genes when you need them. So on your notes, you have a couple of things. So the regulator gene is normally, right here we go, regulator gene is normally outside of the operon and that codes for the repressor. Good. And the promoter is where the RNA polymerase binds. The operator is the region where the active repressor binds, preventing, so when it binds, it's preventing RNA polymerase from binding to the neighboring promoter. And then structural genes, I gave you everything for that. So the lac operon, it's called the lac for lactose, right? The operon is normally off, right? So the lac operon is normally off, and it has an active repressor, but this active repressor can be inactivated, right? And if we inactivate the repressor, then the operon is on. So when only lactose is available, then the structural genes that code for the 3 enzymes required for lactose metabolism are produced. The lactose binds to the repressor and inactivates it. And lactose acts as an inducer. That's why it's called an inducible operon, because its presence is required in order to express a gene. And this is very typical of catabolic processes. Remember, anabolic processes are about building and catabolic processes are about breaking. And what are we trying to break down? Lactose. Let's not make the enzymes unless we have the lactose around that needs to be broken down, right? So that's a very elegant process. Okay. Now, part 2, we're going to talk about eukaryotic regulation. Now, remember, eukaryotic cells, we don't have a single circular chromosome. We have multiple linear chromosomes, and they're contained within a nucleus. So it's going to be a little bit different. Okay, so reminding you again, right? Transcription, translation. All right. Hopefully you remember that. We went over that earlier. So underneath your control of gene expression for eukaryotes, most cells have complete complement of DNA, all the same genes. And we talked about this at the beginning. Different genes are turned on in different cells. So let me show you what I mean by that. And that is called differential gene expression. Differential gene expression. So here's a red blood cell, muscle cell, and a pancreatic cell, okay? Specialized, right? Now, they have housekeeping genes, let's say for metabolism, that are always on, okay, by the blocked out. That's the gene that's on and getting transcribed and translated. But in red blood cell, the hemoglobin gene is on, but it's not in the muscle or the pancreatic cell because those two cells don't need to make hemoglobin. They have the gene, but it's not turned on. Whereas the pancreas is in charge of making insulin, so it turns on the insulin genes. The myosin is important for the muscle cell, so it has these genes are turned on. So the same DNA in all the cells, but different genes are turned on and stay active in different cells. So the ones that are always on are called housekeeping genes. All right, so let me make sure I've got you on your notes. I think we have it. Yes. Okay, so there are 5 main levels of control that you're going to need to be able to know and differentiate between. So here they are. I have taken a picture out of your book and I have annotated it a little bit. So the first level, and I'll go into the details of this, the first level of control is chromatin structure, the state of the DNA. Is it unwound or is it tightly wound and put away? The second level of control, this is the most critical level of control. This is transcriptional control. Can RNA polymerase get on the promoter and make that mRNA? Post means after. So after transcription, what do you have? Do you remember that pre-mRNA? It needs a cap, it needs a tail. And remember, we cut out the introns. So anything that influences that, cap, tailing, cutting out introns, that is post-transcriptional control. And it also includes the ability for that mRNA to even leave the nucleus, or is the door shut? Okay, translational control, that's number 4. That is anything to do with how long the mRNA lasts, whether or not it can get on the ribosome and be translated, and that you can be putting together that polypeptide chain. And then the last, post, remember, means after. So post-translation, you usually need to fold your protein into its correct shape. Okay, so we're going to talk about those 5 levels of control. So, let's see, chromatin, okay? So eukaryotic regulation, chromatin structure. So we'll start there. So I want to remind you, and we've talked about this a little bit already, but when you look at a chromosome, right, when it's in this shape, that means it's going to go through mitosis or meiosis, right, when it winds up that way. But the DNA itself is wound on normally these histone proteins. Histone proteins are non-regulatory, and 8 of them together form what's called a nucleosome, and the DNA is wound on that. Now, if the DNA is wound up, tightly put away, then it cannot even, you don't even think about RNA polymerase getting on a promoter or anything getting in the way of that, because that DNA is all coiled up. If it's open and unwound, then it is available for RNA polymerase. So the first level has everything to do with what's the state of the DNA. So the more tightly wound on the DNA on the nucleosome, the less accessible it is for transcription. So I'm not going into the details about this here in honors bio, but it has to do with methylation and acetylation. So methylation, it puts it away. If you get a acetyl group on one of these histone tails, then it tends to unwind it. Okay, that's just a little more detail on that. So on number 1, underneath chromatin, DNA is wound onto histone proteins. The more tightly wound, the less accessible it is for transcription. The less accessible it is for transcription. And so here you can see this DNA methylation when a methyl group is on there, it's tightly coiled and put away, and acetylation will help unwind it. But this has to do with something called epigenetic mechanisms. Do you remember how we talked about when we studied Mendel, right, that your DNA codes for your traits? But if there's something above that influencing whether that gene is even available or not, then it doesn't matter what the DNA says if you can't even access it, right? So that's above genetics, epigenetic inheritance. And these can be influenced by things like development, environmental chemicals, drugs, pharmaceuticals, aging, your diet. Can influence what DNA is even available to give you the traits that you have. Now, when you look at a nucleus, so here this darker line right here is the nuclear envelope, right? Inside here is the nucleus. This is the nucleolus. You can stain it, and the dark staining one, this darker purple right in here in this picture, that's called heterochromatin because it looks different than the other stuff in there, right? And heterochromatin is dark staining, and it is not active. And the reason why it stains so well is because it's coiled and condensed, whereas euchromatin would be this light gray in color here. It's non-staining because it's all unraveled and available, and that's the kind of DNA that is getting actively transcribed and then eventually translated. So on your notes, heterochromatin, dark staining, inactive, and euchromatin, light and don't stain in active genes. Examples of heterochromatin, you have all that in your notes, is what's called a bar body. Now, women, females, biological females have 2 X chromosomes, right? A male has an X and a Y. And males do just fine with a single X, right? Now, women have 2 X chromosomes, so that's double the dosage, okay, of that X chromosome. So what happens to keep the cell in homeostasis is one of those X's condenses and it forms what's called a bar body, okay? And that is an example of heterochromatin. And in a female cell, when you look in, you can see that the different X's could be condensed in each cell. So that's why you could have identical twin girls have different colored eyes or other differences depending on which X is condensed in their cells, right, for whatever traits they control. So another example of heterochromatin, oh, here I've got another picture for you. Here's a bar body inside of a nucleus, and also right here. That's just an X chromosome that is condensed as heterochromatin. Now, another evidence of that is if you look at calico cats. So the coloring on them depends on which X is available for transcription and translation, and that's why you get that mottled color. Now, my question, riddle me this, do you have male calico cats that look like this? And hopefully you're telling me no, because they only have one option on their X. So it is whatever it is, because that's the only one that gets expressed. Okay, so I gave you all the notes on that. All right, next level of control is transcriptional control. And this is the most critical level of control. Now, let me get that. There we go. This is a simple diagram, but I just wanted to remind you what we learned about transcription just a few minutes ago, right? So RNA polymerase wants to bind to the promoter, right? And there could be a repressor that gets in the, there could be a repressor that binds to the operator, which then inhibits RNA polymerase from binding to the promoter, right? And that is typical of prokaryotic cells. Prokaryotic cells are all about repressors and whether they say no or not, okay? Whereas in eukaryotic cells, that's not how we function. We're not so much about the repressors, we're about encouragers and things that assist RNA polymerase to bind to the promoter. And those, the common one I want you to know is about transcription factors. So here you can see the promoter. It's right here in green. Here's RNA polymerase. There's no repressor in the way. that would inhibit RNA polymerase from binding the promoter, but it needs help. So what you have is you have transcription factors that will assist RNA polymerase to bind to that promoter. And it's not just these transcription factors, but farther upstream, can you see where it says transcription activator? Now, I know that looks like lactose, but it's not. So these transcription activators bind to another region called an enhancer, and it forms this hairpin loop, which then helps the transcription factor help RNA polymerase to bind to the promoter. So we're more about, do we have the helps or not? We don't have operons. We have multiple genes, right, located on multiple chromosomes. So the way we can control them together, is whether or not we're assisting in that process of transcription. Okay, now let me move myself right here. Okay, so sorry, I had to get the phone for a second. Let's make sure we have the notes on that. All right. So underneath transcriptional control, you're going to put the most critical, right? Most critical level of control. And you have transcription factors. These are proteins that assist RNA polymerase to bind to the promoter. That goes on your notes. And then transcription activators, I gave you everything for that. They're usually farther upstream where they will bind to the enhancer region and then help the transcription factors to bind. Now, examples of transcription activators and transcription factors, those can be seen in cell signaling. So let's take a look at this, all right? So here you have a signal molecule. It's binding to a receptor, right? And this triggers a signal transduction pathway like dominoes, which then ultimately makes this transcription activator functional. And then it can bind to the transcription factors to turn these genes on to produce this mRNA and eventually translate this mRNA into a protein. Now, this is a very, very simple picture of cell signaling, because in reality, it looks like this. Okay, so you can see different receptors that are here on the cell surface. This is part of that signal transduction pathway, influencing which genes are ultimately transcribed and translated. All right, so your example is cell signaling. All right, next, post-transcriptional control. You remember, because we've studied transcription, what has to happen after you form your mRNA, right? For post-transcription, let's take a look. I want you to get your bearings up here, because there's more to this picture than meets the eye, and it's a good review for you. So look in the top left-hand corner. We can see the DNA double helix. You can see it's unwound, and your RNA, now this could be mRNA, tRNA, or rRNA, right? You can make all of the different kind of RNAs, there we go. But let's look right here for now at this pre-mRNA, and notice it has its introns still in it. So post-transcription has to do with, what are you going to do with that now that you have that mRNA? Well, you're going to cut out the introns and then splice together your exons. You're going to put on a cap and a tail, and then that finished mRNA needs to be able to leave the nucleus. So all of that is post-transcriptional modifications, okay? And we need to keep these straight. Transcriptional control is the making of the pre-mRNA, right? And then post-transcription is cap, tail, and cutting out the introns and leaving the nucleus. But I also like this picture because it shows you the other RNAs. So here you can see tRNA. This could have been tRNA that we made, which needs to then get charged with the correct amino acid, right? So it can go to the workbench. And then our RNA needs to hook up with some proteins to form the small and large ribosomal subunit, which together here look like a turkey, okay? But this is where translation will take place. So on your notes for post-transcriptional control, differential excision, removal or inclusion of introns, and splicing. Okay, splicing means linking, and the speed of the transport of mRNA through the nuclear pores. Also add to number 1, adding in the cap and the tail, putting on the cap and the tail. All right, so here you can see different mRNA processing once again. So we can see the cap, the tail here, we can see exons A, B, C, D, E, and their introns between them. In this splicing, they've removed the introns, and you have A, B, C, D, E, and you have this protein product 1. But let's look at scenario number 2. Cap, tail, cutting out the introns, but what else did they remove? They removed C, one of the exons. So as a result of that, you're going to have a different protein product, but it's coming from the same DNA, right? So depending on that differential excision of different introns and exons, you can get a different mRNA, which will give you a different protein. All right, next, let's talk about translational control. So translational control is anything to do with the mRNA being able to get onto the ribosome and be able to build the polypeptide. So how long the mRNA lasts, if there's any small interfering RNAs which influence and turn off that mRNA. And I don't want you to think of gene regulation like, darn it, you're not letting me do this. It is part of differential gene expression, turning on the right genes for the right amount of time to make the right protein product. So translational control, you have all of that one, okay? And then post-translational control, okay, is after you've made that polypeptide chain. Like you can see this polypeptide chain, it's not done now. Okay, you made the chain, but it's not functional until it is folded into the right shape. Sometimes you have to remove some of the amino acids, and sometimes it needs to be folded into its correct structure. Do you remember when we talked about the 4 important organic molecules? There was primary, secondary, tertiary, and quaternary. Primary was the sequence of the amino acids. The secondary structure was either an alpha helix or a beta pleated sheet. The tertiary structure was the folding pattern imposed upon the secondary structure due to the R groups, of which there are 20 different ones, and the quaternary structure is if there's more than one chain. So that tertiary quaternary connection, that is post-translational control. So following translation, only a functional protein is an active gene product. So here you can see, here are different genes. Here are the RNAs that are getting made, right? So if the DNA is even available, whether it's heterochromatin or euchromatin, that's control 1. Whether or not you can do transcription, that's control 2. Post-transcriptional control, introns and exons, cap, tail, and leaving the nucleus. Translational control is if you're able to build the polypeptide chain, and post-translational control is if you can fold it. Okay, so hopefully that is a good review for you. And in video 2, I will talk about failure of control, and that is called cancer. And if you're one of my students, I'll see you in class.